Good morning, everybody. My name is Michael Crawford. I'm the Chief Executive Officer of Love of Soil CRC. Welcome to today's webinar on how farmers can, on affordable rapid field-based soil tests. I'd like to begin by acknowledging the traditional custodians of the land upon which we meet today, wherever we are, and I pay my respects to our elders past and present. I extend that respect to Aboriginal and Torres Strait Islanders peoples who might be on the webinar today. Today's webinar is an update on some research in Program 2 of the Soil CRC. Program 2 is around soil performance metrics, basically around what to measure, how to measure it, and what to do with all the data. In terms of how to measure it, this project that you're about to hear about is consistent with our objectives in this space about how farmers can measure critical soil performance metrics quickly, cheaply, and accurately. A few words about the Soil CRC, appreciating that many of the attendees already know quite a bit about the Soil CRC, but we have many visitors both from within Australia and internationally. The Soil CRC, the Cooperative Research Centre for High Performance Soils, its full name, is essentially a collaboration of 40 participants across Australia and New Zealand, including 8 universities, 4 state government agencies, a number of industry groups and associations, and importantly, 20 farmer groups or grower groups. And through these grower groups, working with researchers, that we're able to achieve what we're trying to do in the Soil CRC of ensuring that we can undertake soil-related R&D to give farmers the tools and knowledge they need to improve their soil management and ultimately their productivity and profitability. The Soil CRC has funding from the Australian government and from its participants for 10 years. We've just commenced in the 4th year of the Soil CRC. Today's webinar is on, as I said, affordable rapid field-based soil tests to be delivered by Dr. Liang Wang of the University of Newcastle and Reuben and Yang, a PhD student at the University of Tasmania. And this is indicative of all our projects, and they represent collaborations between universities, research partners, and you'll see in a minute between farmer groups as well. Just whilst we're here, we've been running a program of webinars every fortnight or so since June. We've got one more webinar left this year on the 8th of December to be given by Dr. Lucas Van Sweeten of New South Wales Department of Primary Industries. Lucas is also the leader of Program 4 in the Soil CRC. And the focus of that one is soil biology under the microscope. And just an important note, that will be delivered at 12 noon rather than 11 AM for traditional time, 12 noon Australian Eastern Daylight Time. So to come back to our speakers today, Liang Wang, before I go there, just a couple of housekeeping items. We are recording this webinar and we will make that webinar available on our website in the next couple of days, next day or two, so you can go back to it afterwards. You can also link to it and share it with your colleagues should you find that worthwhile. In terms of questions, the way to ask questions is to type them into the Q&A box, which you'll see at the bottom of your screen in the pop-up there. Type in the questions as you go, and at the end, we'll take about 15 minutes or so to go through those questions that are there and provide the answers. You can write other comments in the chat function if you like, but put the questions in the Q&A box. So, our speakers today, Liang Wang is a Senior Research Fellow at the University of Newcastle in New South Wales, Australia, at the Global Centre for Environmental Remediation in the Faculty of Science. His research interest lies in coupling colourimetric computational methodologies to environmental processes and problems. These include rapid determination of contaminants in foodstuffs, coupling of erosion processes with in-situ measurements, and also applying advanced analytical measurements to ecotoxicological processes. Reuben Yang is a graduate in the field of agricultural sciences with honours at the University of Tasmania, graduating in 2018. He's originally from Malaysia. He's now pursuing a PhD at the University of Tasmania in conjunction with the Soil CRC with Source CRC support and part of his team of scientists in developing rapid and affordable in-field devices for analysis of soil nutrients. So without further ado, I'll pass over to Liang. Liang will speak first, and I'll pass over to Reuben, back to Liang, then we'll open up for questions. Thank you.
Good morning, everyone. This is Liang. Today, Reuben.
Liang, we'll just move back to the screen share. So the viewer, you are sharing your screen, but not the viewer mode. Not the presentation mode, if you know what I mean. That's the one. Wonderful.
Okay. So, morning, everyone. Thanks for Michael to introduce us and fund us with this wonderful, interesting project. And today, me and Reuben, we're going to introduce our project. Title is Affordable Approaches to Rapid Field Testing of Soil Chemical Properties. Our project team, we have two universities, University of Newcastle and University of Tasmania. And we also have two industry partners, Reuben Productivity Service and King Productivity Service. We have two PhD students working on this project, Reuben and Sebit. They're all based at University of Tasmania. Project aim, so now is the revolution of agriculture. People are more interested in using a rapid soil sensing system and also developing a simple and affordable solutions for farmers to test their soil plants in field. Same as our project, we are aiming to develop solutions for farmers to determine soil chemicals, properties, and we try to tick all the boxes, including affordable, easy to use, and has to be bring to the field, get the rapid results, and the results should be available and more accuracy and reliable. We start with some background with soil chemical properties. First, most important is the pH. Soil pH is very important for plants, and most of the plants can tolerate in a very narrow pH range, which is neutral from 6 to 7.5. At this narrow pH range, plant can access most of the nutrients. When pH get lower, soil get acidic, and we cause and reduce soil microbial activities, which cause major nutrient deficiency. When pH get higher, soil get alkaline and cause the metals like iron and other trace elements getting deficient. Primary micronutrients, we all talking about nitrogen, phosphorus, and potassium. If we are growing green vegetables like lettuce, spinach, then we need more nitrogen. If we need to get flowers, fruits, and roots like tomatoes, carrots, potatoes, then we are talking about more phosphorus. Potassium is for overall, for more healthy of the plants. And different plants definitely need a different ratio, a combination of NPK. But unfortunately, most of the soil don't have that, all the nutrient one plant needs. So that's why we need to add supplements, fertilizers. Different products have different combination of products, this ratio. But how can we select the right product? We need to base on the plant and based on the soil condition. First, if we add too much fertilizers, first of all, fertilizers are expensive. And if we add too much, it's gonna kill your plants. And also if we're doing over-fertilization, can lead to long-term soil degradation, including deterioration of soil fertility, damage the balance of the current elements. If add too much potassium, it's gonna increase the soil salinity and solidity. If we're adding too much acid-forming nitrate fertilizers, it can cause decrease of soil pH. As mentioned, if the pH get lower, it will reduce the activity of microbials. and also cause deficiency of other micronutrients. And the remaining fertilizers can be carried out by runoff to the downstream and also go downward to contaminate groundwater, cause eutrophication. So before and after we adding fertilizers, we should measure soil conditions. The most reliable way is sending the soil samples to the lab, but it's costly. Normally per sample, we charge us about $50 to $100. And we have to wait for a couple of days to get the results. And in this circumstance, we can't add faster. For example, if we add too much fertilizers, how do we know? How can we react as quicker as possible? We can't wait for a couple of days. Another solution, alternatively, we can use the soil field testing kit, which normally comes with a case that bring all lab things to the field. And they provide all the chemicals, it's called color reagents. When we prepare soil solution and we add color reagents, the reagent will interact with the nutrients or elements and create indication color. Then they provide a color chart to compare and to indicate the level of the nutrients. But unfortunately, most of the soil are colorful and the soil solution will have the background color. If it's transparent or no color, this method can work better. But this background soil solution will add interference for your readings. So this method can only give you approximate results. If you want to know NPK ratio, it's very difficult. So our project goal is to produce a single device to measure all the micronutrients and provide you more reliable results, and can be affordable, user-friendly, and farmer or end user can bring it to the field and measure and get the result rapidly. We have two activities. One activity is to develop the hardware, which is small size device, pretty much like a fingertip size, to contain all the color reagents to measure all the major indicators, NPK and pH. In that case, you can bring a dozen of them into your pocket. You don't have to bring the suitcase. Second activities, instead of using your vision to tell what's the color and indicate the color level, we use camera, your phone camera, to get the results, to get the color, RGB colors, out from the device and interpret that to concentration. Now let's go to more details of each activity. The first activity is the hardware development. We more focus on 3D printing microfluidics. And our PhD student, Reuben, is the main student focused on this research direction. So I would like to hand over to him to give you more introduction. Reuben?
Thanks, Liang. Ladies and gentlemen, I hope that by now you would have understood how important soil analysis is to the productivity of the farmland. With the lack of real-time results from laboratory analysis and the lack of accuracy from current in-field devices, the lab on chip as we present today is the way forward, which combines the best of both worlds. Next, please, Liang. The lab on chip concept, known as microfluidics, involves the manipulation of fluids that are constrained to a small scale, which has benefited the scientific community due to using less volume sample. It's fast, it's easily integrated into experiments and tests. The biggest cost advantage of microfluidics is that devices can be easily produced with 3D printing, which in our project is used to bridge the gap between microfluidics and agriculture. Simply because 3D printing techniques provides endless opportunities, such as rapid design iteration, allowing you to develop devices and prototypes, as well as modifying them on the go. Materials are often strong, cheap, and easily available. It's mass producible, disposable, and again, devices and prototypes can be produced on demand right there and then via desktop production. Therefore, Our project is to develop a handheld device to rapidly determine the soil nutrients that reside within the soil right there and then, right in the field as we speak, where the soil sample undergoes a filtration within the device and a reaction with an analyte-specific reagent to produce a color. The intensity of this color reaction or the color formed will then be correlated to the concentration of the target analyte, which finally it is quantified using our mobile phone camera software application. Early device prototypes were developed by Ms. Sepide via photolithographic printing techniques, which consist of a reagent chamber, a filter membrane in the middle, a sample chamber, and finally a sample input cavity as shown in the image. The flow dynamics of the device include, firstly, injecting a soil sample into the sample chamber where the sample flows through to the filter membrane, in the middle where it is filtered, and finally reacted with the reagent in the reagent chamber shown in the magenta red illustration. And finally, this color is then converted into numbers with, again, our mobile phone camera application. Our team at UTAS have carried out developments in the filter membrane of the microfluidic device, which can be easily printed using photolithography. We did realize that changing the print angles would hence alter the porosity of this filter membrane. Upon developing this filter membrane, we proceeded to carry out tests using various particle sizes to understand the filtration efficacy. These are images from under the microscope that shows that the filter membrane is able to filter particles up to 10 microns, as shown in the image on the far right. But given that clay is less than 2 microns, its influence in the results have already been considered and factored into our analysis. This is another example of a test that has been carried out using river samples in the image shown to reaffirm the efficacy of this filter membrane. Simultaneous tests have been carried out as well on the filter membrane using pH as a means of measurement, when we exposed the pH reagent to a soil solution sample and as well as a clear sample to understand how it works. The top row of the image shows a reaction between the soil sample and the reagent requiring about 10 minutes. However, in the bottom row, a clear sample solution on the bottom row only took 60 seconds to react with the pH reagent as shown in the third image. Therefore, the purity of the sample solution will determine the filtration time required. As mentioned earlier, these tests were carried out using pH as an indicative measurement, which also benefits other aspects of our project, which are conducted by the team from the University of Newcastle, which I'll hand it over back to you, Liang.
Thanks, Reuben. Very nice presentation. Now back to me. I'll talk about the second activities, software development. So all about using a smartphone to interpret the color to the concentration. Follow up with his slides. We take the color and interpret that using our algorithms. We have to mention that all this color forming are producing a redox like chemical forming. They are temperature and light related. So light temperature can affect these procedures. For example, if you are doing a measurement outdoor in a cloudy winter morning and compare the same solution in a summertime, sunny day and midday, your color can be different because some of this color indicator is not that stable. And with the hot temperature, it can be changed very quick or precipitate. So our experiments set up in a constant temperature room with constant lights, with the full spectrum LEDs, and we record everything and following how it changed. Then we put it into our algorithm to make sure we get the ultimate results. Furthermore, as mentioned, soil solutions are colorful. We have here, we have 3 different soil samples. On the left-hand side is a high organic dark soil. In the middle is a high clay brown color soil. On the left-hand side is a sandy gray color soil. So we prepare a soil solution after filtration. You can see the difference compared with standard solution. The color, that's the background color, gonna add into your measurement and cause interferences. And our model will, and is doing it now perfectly, to remove this background and give you reliable results. A summary for our software development, we use colorimetric algorithm to interpret color values to concentration, including NPK and pH. Chemometry algorithm to give you improving the prediction accuracy, improving the results by minimizing or reducing the negative impact, including temperature, light density, and background colors as well. Decision making, we will link the prediction results with soil health, provide the end user with information about and help them to make the decision. For example, we can link or cooperate with fertilizers companies and using their product database and provide the suitable product and recommend that to our end users. Summarizing of this presentation, we are developing a solution for farmers to determine chemical properties for their soils. And the prototype will be developed in next year, including 2 parts. One is the hardware, all about 3D printing microfluidics. This fingertip size device can measure all the micronutrients in once, which you can put into your pocket and measure as many as you want. And also integrating filtration systems, which can remove most of the particles. Also, we have a software development using the camera. Instead of using your eye vision to tell the color, we're using a smartphone to interpret the color to the results. And our results will be more reliable because we have sophisticated chemometry and colorimetric algorithm integrate into the system. And also we support your decision by giving you recommendations. And that's the end of our presentation, and you are all welcome to give questions. Thank you very much.
Thank you, Liang. Thank you, Reuben. So, yeah, very exciting developments here in the research, and I encourage you to type questions in. We've already got a couple of questions there in the Q&A box, but I'll just start off, and Reuben, you can show your video again. That's good. I'll start off with a question, Liang, and maybe it's for you, Reuben. But technology is one side of it, which we need to develop scientifically. But I guess the other question is, how might we use this? How might farmers use it in the field to actually inform their decision making? I understand that in the future, once you scale this up and through the 3D printing, the cost per chip, which are largely disposable, or one single-use chips, the cost per chip is quite low. Can you talk about some of that and how you envisage and what some of the feedback has been from farmers to actually use it? So if you're going to answer the question, come off mute.
Yeah, that's a good question. To figure out how to use it will be very similar to the current soil testing kit. We prepare a solution as indication, but that would be very simple. And then we just inject this soil solution into the chips by one channel. They're going to split out into different channels for give the colors. And the results will show in very quick couple minutes. Then using a camera, and they will record the results straight away. And the cost will be the cost right now from our part to develop this product is be very low. So I don't think how much on top will be add up, but I believe that will be very cheap for a farmer to do the simple test.
Okay, Reuben, did you want to add that? I think you mentioned a cost per chip to me in the past. Do you want to talk about that?
Yeah, absolutely. So currently with our 3D printing techniques and the devices given to us, printing a single device, which of a minute scale, would essentially be less than $1. In fact, I would say possibly less than 50 cents. Depending on the size of the chip and also the dynamics as well as the complexity that resides within the chip, it might differ. But at the moment with our development, it's definitely less than $1, close to 50 cents. We are looking at alterations with the current chip prototype, which again, we are unsure of, but in the future, we might come with a finalized design or prototype, which again would finalize the price essentially. Does that answer your question?
Yeah, that's very exciting, and I guess as CEO, I'm allowed to caveat, but there's still a lot of development to research and development to get to that end user stage first. Let's go to some of the questions. First one is from Jim, and others who are looking at the Q&A. If there's a question there that you really want to see answered first, you can give the endorsement the thumbs up, and it'll go up the list. But from Jim Laycock, how does soil moisture affect the result? Liang, come off mute.
Soil moisture probably wouldn't affect the results because we prepare soil solutions with water. So I'm not sure how much percentage of soil moisture gonna partially add into the solution, but I don't think that's a problem from the point of view currently I can see. Thank you for the question.
Okay. From Steve Kimber, make a comment, but you can take it as a question. Presumably, accuracy will be increased by conducting classical analysis on a subset of samples. Year-on-year testing should inform the system. Yes, I guess how would, thinking to the future, how might these chips be used in conjunction with classical or standard laboratory testing? Would it be complementary or as a substitute?
At the beginning, maybe we can compare if you measure that with our system, and then you send duplicate that and send to a laboratory to get the results and see how it's correlated with the results. And then when you're doing a large scale measurement, you might just pick up randomly, select a couple of samples and send to the lab to do the validation. That's the recommend I can provide.
All right, thank you. Peter Harrison's asked about the likely cost for the system. Now, I think we might have addressed that in those opening comments, unless you've got any other additions. No, okay.
No, I think Reuben already mentioned the cost from our side. Yeah. Not sure how much you're gonna add up because this commercialization, even you have a $1 cost from your side, but advertisement, everything gonna add up. So it's beyond our control, so we can't answer that question, sorry.
Yeah, and recognizing the difference between cost and price. Okay, Steve Kimber has asked, would it be better to incorporate a commercially available QC produced filter fits in the chip, I don't know, yeah. Strip maybe, yeah.
It's a good question. Yes, if we're using an additional external filter that will have better removing the particles and definitely giving a better results. But what we're aiming to do is to lower the cost and integrate the filtration system into the 3D printer. But if we have that in additional, that'd be great. But not sure how much cost we're gonna add on.
Okay. Jeffrey Crack from Fertilizers Australia. How do the measurements from this technology relate to plant growth measures and responses?
Thank you. I'm not the knowledge. I don't have the knowledge about that. But just saying that we measure the soil. I give you the NPK ratio, and based on your plants, you definitely need different ratio of that. Then you can, it might be other experts and can get the knowledge into our app and link to a database and provide related information. It's extendable, so so definitely a lot of potential there. Thank you.
Yeah, thanks. And I think I'd add to that for Liang and for Jeff's it's a topic worth discussing. But I think, what this technology would do was give you a measure, and then how you make a decision, how you respond to that, what level of fertiliser you use in addition to that, how the plant responds, will be part of the ongoing further development, the agronomic aspects of this technology as we start to make it field ready. Thanks, Jeff. From Richard Doyle, who's a program leader in Program 2 from the University of Tasmania. Thanks, guys. Very interesting and exciting. Can you discuss the ideas about the source sample handling to introduce it into the device?
Thanks. We are basing on the procedures as normal will be prepared for other like commercial available products. We follow the same procedures. So we don't think we do any additional on top. So we just try to make it easy to use, user-friendly. So if there's no special additional filter add in, if that chip, the device work well, I think that just by proportionally adding soil and water to mix up. Thanks.
All right. Thank you. From Penelope Cook from Penny Cook, great presentation. Will there be a way to store results so a grower can enter those into their farm paddock records when they are back in their office?
Yes, the application not just limit to the phone app. Then you can use you can integrate into other automatic systems, like you have a camera or something. Then on the machine, then you can automatically measure or make it everything automatically, and then you get the results, send the digital data back to your office or database or into the cloud or online. Everything's achievable.
I think that's right, Liang, isn't it? Once you're using a smartphone or that sort of technology, then the world's your oyster in terms of how you can use that data and connect into the whole range of apps, both mobile and back in the office.
That's right. We can put a database online.
Good thinking, Penny. From Jim Laycock, how is it critical value, for example, phosphorus, determined with colorimetric measures?
Sorry, how's the phosphorus?
So taking phosphorus as an example, how do you determine the critical value with colorimetric measures?
Okay. I'm not sure how do I answer this question in a different angle, but phosphorus definitely is very important, and it really depends on how accurate our results gonna get, but definitely will be much better than the current field testing, in-field testing results, which just give you approximate, but we definitely give you a better range, and then you can base on the ratio of what you need, and then because that we need other additional knowledge about what plants you're gonna grow on the soil, which the soil conditions as well. So based on what the lots of information, then we can tell that you should add additional, or what you have to add is over. So that's what our can potentially suggest.
Yes, yeah, okay. I think that makes sense. And given the critical values are dependent on soil type, sand, clay, lime, etc., then plant crop type, as you say. Yeah, similarly with lab tests, correlating with those lab test results to determine those critical values will be important. Richard Doyle has asked another one, can the smartphone also locate the sampling point and the ambient temperatures? Can a moisture scanner be provided via the smartphone? A project there, Rich.
But there's lots of potential. So we not try to develop something duplicating the current market available products. So for example, this sort of a moisture sensor is something already be developed and connect to your smartphone. I think that's very simple to achieve, which is not a research project. So we can integrate that. So GPS location is simple. So you have that straight away from your phone. So all this additional information definitely can add into the app to help to make the decisions.
Okay, thank you. And Peter Harrison, on a similar theme, how will record keeping be organised and records kept on phones? You might have touched upon that already, Liang, but have another go. Peter Harrison's question.
The data can save in your, you can either way, so save in the cloud or save in your phone or other computers, that all can be achievable. So just technology as tech, so it's not a big deal. So.
Chris Gazy from West Australia has asked, a big question with soil testing is a collection of a representative soil sample. At this stage, do you have information that growers will adopt this technology? Very interesting work and great presentation, thank you. And I think it's a key issue about, if you've got this sort of technology and we recognise the limitation or the issues, but the importance, sorry, of representative soil sampling. We know that from traditional testing, how you essentially get a number that represents, what's happening in a 100-hectare paddock. Yeah, how do we get a sample that represents what's happening in a 100-hectare paddock when you've just got essentially a drop of soil in a little chip? I think there'll be some next stages are quite interesting on the statistics, on the biometrics of what's the best way to do that. Is it multiple sampling or multiple testing versus bulking and subsampling type approaches? But Liang, maybe you've given some more thought to that too.
Thanks. Yeah, that's what we're aiming for actually. When you make the measurement cheaper and you get the results much rapidly, then you can measure much more than current what we're doing. So instead of just one or two selecting location, you can do it in a grid. That's what we're doing for soil mapping. So select as many as you want. If you have time, do a week and 100 samples. Then if not, there's 20 or dozens of samples. It can give a better profile of your soil rather than just one or two samples. So that's what we're aiming for, to make the cost cheaper and then give you a rapid, faster results.
I would like to add on to Liang's comment.
Reuben.
I'm sorry, I'd like to add on with Liang's comment. Also, because our device at the moment is quite small considering its size, so I would say that it would provide the farmers a more versatile method of testing, essentially, rather than to have to bring soil samples back to, for example, his house before they can be sent off to labs. He could essentially just take a small sample of soil, do it right there and then, get it into the chip, and then get his color reaction in about a few minutes' time. And most of all, getting as much samples within the paddock would essentially have a more robust system of measuring your soil nutrients there and then.
Thank you. And in fact, and Steve Kimber's added to Chris's question, or partly answered it by saying, potentially use this system to plan out the sampling strategy before taking samples, which then go to the lab. So you can start to work out the variability and diversity within the paddock and then subsample accordingly. Frank Agbella from the University of Newcastle has asked, great presentation, following from Richard's question, is there room to measure precipitation, wind, humidity, soil moisture, etc. at a time when the farmer is measuring soil chemical properties?
Yeah, well, as mentioned, that wherever the product available in the market, if it works well, then we don't have to do something from there. And then definitely the data from that sort of product can add on benefit for better results from our view and give you more comprehensive reading for your soil profile. But what we focus on is the chemistry or chemical part of the soil. So we do what we can do, and definitely getting more information definitely would be much better.
Thank you. And just to finish here, so if there's any other questions that people want to ask, quickly type them in. Richard, the program leader, has made a comment on one of those earlier questions about critical values, and rightly said that the work on critical values will follow, like for any new soil test, once we get the range and responses in place. So without seeing any further questions come up, I'll start to wrap it up. And so thank you, Liang. Thank you for Reuben. Obviously, well, in my opinion, a very exciting piece of technology. Okay, so Michael's just come in for question. You might see it there. Different cameras sense colours differently, and it depends on ambient light and time of the day. How are we adjusting for that?
Oh, you pick up the key question. Yes, that's what the algorithm or our modeling for. And then we definitely will have in situ calibration for different the camera or your smartphone or in situ calibrate to give like color adjustment. It's not just based on what we calibrate, it's based on your phone. So thank you.
Okay, thank you for the question and that answer. All right, so I will wrap it up now. Thank you very much, Reuben and Liang, for that presentation. The technology is very exciting. Potentially, when you think through the possibilities that having a little chip at less than $1 each, being able to get, well, obviously cheap, rapid, and fairly accurate, depending on the purpose, technology in the hands of farmers, can really start to change the mindset or the way people might collect soil information and make decisions based on that in a timely manner, rather than having to wait a week or two for getting results from the lab. It might lead to more refined soil testing, but it might lead to a greater adoption of at least a basic level of soil testing, soil information collection, and more precise management of what's happening within a farm, within a paddock. So obviously a great amount of work yet to be done in terms of taking it right through to a commercially available stage, but through the Soil CRC and with the input of the University of Newcastle, University of Tasmania, and other partners, including our grower groups who are there giving feedback on its sensible use from a farmer perspective. We look forward to be able to bring this sort of technology to market in the coming few years with appropriate partners. So I'll leave it there. Thanks everyone for your time. Thanks again, Liang, Reuben. You'll be able to view this webinar online in the next day or so and beyond. Thank you all.